Hi, everyone. Welcome to the Bettersize channel. This is Paddy. Today I'm going to introduce our brand new instrument that is used to analyze stability of dispersions. The instrument is called BeScan Lab and it applies SMLS technology, namely static multiple line scattering, to capture the signal variations during measurement that are used to characterize stability. In this presentation, first, I will talk about the definition of stability and the factors that affect the stability. And then I will show you the BeScan and its measurement principle. And the next topic is the results analysis. In addition to stability analysis, our software also provides many functions, such as phase separation hydrodynamic analysis, temperature trend testing, and mean particle size measurement. In the last part, I will share some application cases with you. The first part is about the definition of stability. So, what is stability? In general, stability refers to the ability to resist changes. However, people have different understandings of the concept of stability. For dispersions, the stability that I'm talking about is kinetic stability. Dispersions like suspension and emulsion are thermodynamically unstable systems because small suspension particles or emulsion droplets tend to decrease the large specific surface area and surface energy. In manufacturing, in order to increase the kinetic stability of the product, various precautionary measures are taken, including the addition of stabilizing agents, adjustment of pH value, particle size control, and homogenization. Therefore, it is possible that a dispersion is thermodynamically unstable, but kinetically stable. Various formulations are screened, in which a stability analyzer is used to evaluate their kinetic stability. A good kinetic stability over time indicates good performance of the product. Whether the product is stable or not is defined by the users. For example, if a mighty unstable phenomenon, like slight sedimentation, occurs and the users believe that it is acceptable, the dispersion is considered stable. If the criteria for judging stability is strict, the dispersion is considered unstable. If dispersions are unstable, in most cases, unstable phenomena can be observed with our naked eyes. Here are some examples. The samples in the figure are suspensions and slurries. After placement for 60 hours, sedimentation and breaking occur. What if unstable phenomena cannot be detected with our eyes? Or what if we don't have too much time to observe the unstable phenomena? In this situation, BeScan Lab can help us. Before I talk about BeScan Lab, first, we should have an understanding of typical unstable phenomena of dispersions and the factors that affect its stability. In this slide, I will show 7 typical unstable phenomena. The first phenomenon is sedimentation, where the particles settle down over time. The second one is creaming, which means that the particles flow up over time. Flocculation and coagulation are related processes. They both refer to particle aggregation and are often used in wastewater treatment. The difference is that flocculation is reversible aggregation caused by weak physical interaction. In this situation, particles are loosely bound, while coagulation refers to an irreversible aggregation. In wastewater treatment, suspended particles stick together due to neutralization by adding coagulants. Then, flocculation enables the forming of large and visible flocks. Finally, large flocks settle down due to gravity force. The next phenomenon is coalescence. Coalescence often occurs in emulsion, where 2 or more droplets merge together. Coalescence is an irreversible process because the film is destroyed. With the development of coalescence, breaking may occur, which means the suspend phase and continuous phase are separated. Phase inversion occurs when the hydrophilic-lipophilic balance of the surfactant changes. which can be affected by external conditions, such as temperature and a pH value. The last phenomenon is ostowa ripening. It happens when the smaller droplets first dissolve and then deposit on larger droplets, because larger droplets are beneficial to a thermodynamic stable system. Stability of dispersions can be influenced by several factors, including interparticle force, steric hindrance, particle size, and density. The DLVO theory provides an explanation of how the interparticle force changes as the distance between them changes. The upper part of the curve represents the repulsive force, and the lower part is the attractive force. The red line represents the electrostatic repulsive force between particles, and the green line represents the van der Waals force. The blue line is the net potential, which indicates the sum of the attractive and the repulsive force with distance. As shown in the figure, when the particles come close, the attractive force dominates first, resulting in a reversible flocculation. Then, repulsive force dominates as the particles come much closer. In this situation, an energy barrier occurs, which is a critical factor affecting the stability. If the kinetic energy of the particles is larger than the energy barrier, they can cross the barrier mountain and will be trapped into the area where attractive force dominates again, resulting in irreversible coagulation. If the kinetic energy is smaller than the energy barrier, the repulsive force between particles ensures stability. On the right part of the slide, different net potential curves are shown. The curve is affected by many factors, such as the particle surface potential and the salt concentration. If the surface potential is high or the salt concentration is low, the energy barrier is high. Therefore, the dispersion is stable. In contrast, if the surface potential of the particles is low or the salt concentration is high, the dispersion is quite unstable, and the particles easily aggregate. Steric hindrance reduces the possibility of particle aggregation, increasing the stability. When it comes to particle size, small particle leads to stronger Brownian motion, which reduces the possibility of particle aggregation as well. High zeta potential ensures repulsive force between particles. The density difference between the suspended phase and the continuous phase leads to creaming or sedimentation. Other factors, such as viscosity, temperature, and a pH value can also affect the stability of dispersions. So, why do we need to analyze the stability? Because stability plays an important role to product performance. In the development stage, formulation screening is carried out to search for the most stable dispersion. In most cases, good stability refers to good performance. For example, in agrochemicals, good stability is required to ensure homogeneous dispersion of emulsion droplets. Uneven dispersion of emulsion droplets may reduce its effectiveness. We also need to find out the optimal transportation and storage conditions in case the products are not stable or invalid after long-term storage or transportation. Good storage and transportation stability increases the product's shelf life. Many products need to be shaken or diluted before use. Therefore, it is quite important to analyze the dispersion to observe how the pretreatment affects the stability. If the dispersion is not stable after pretreatment, the formulation needs to be screened again. The second part is the measurement principle of BeScan Lab. Here I present a simplified interior structure of BeScan Lab, a setup containing an LED light, a forward detector, and a backward detector that moves upwards to capture the transmit and the backscatter signals every 20 micrometers. The technology utilized in the BeScan Lab is called static multiple light scattering. It is an optimal method to characterize the sample stability. The measurement is non-destructive, fast, and simple. For concentrate samples, the transmit signal is hardly detected. Therefore, the backscatter signal is applied for stability analysis. The backscatter signal is related to the transport mean free path of photons. If the sample is clear or transparent, we can use the transmit signal. The transmit signal is related to the mean free path. Mean free path and the transport mean free path are related to the size and the concentration of the suspended substance. According to the last slide, we know that the transmit and the backscatter signals are related to the size and the concentration. Here I present the change curves of transmit and the backscatter signals with different volume fractions. In low volume fraction, the transmit signal is easily detected. An increase in volume fraction leads to a decrease in the transmit signal. Finally, the transmit signal is hardly detected because the light transmission is blocked. For concentrate sample, the backscatter signal increases as the volume fraction increases. After reaching its maximum, the backscatter signal decreases because of the interference between light diffusion arising from particles. In this slide, I will show the change curve of transmit signal with different particle size. The volume fraction is fixed. An increase in particle size leads to a decrease in the transmit signal because the transmission will be blocked by large particles. In a certain range, the transmit signal is hardly detected. However, the number of the particles decreases as the particle size increases when the volume fraction is fixed, and the interparticle space increases, which allows the light transmission again. Therefore, the transmit signal increases again. In contrast to the transmit signal, the backscatter signal first increases and then decreases. It is easy to understand when the particle size is small. The backscatter signal caused by the suspended substance is hardly detected because most of the light passes through the sample. As the particle size increases, backscattering increases. Finally, the backscatter signal is hardly detected again because the number of the particles decreases, as I mentioned in the last slide. In the software, the signal will be displayed in two forms, namely raw data and delta data. The raw data refers to the originally detected transmit and backscatter signals. Delta data is the subsequent signal minus the reference signal. The signal obtained by the first scan is applied as a reference signal. With the delta data, we can observe how the signal changes. The x-coordinate is the height of the sample cell. I will use sedimentation as an example to demonstrate how the measurement works. The signal will be present in the form of delta data. As mentioned before, a setup containing an LED light source and two detectors move upwards. The transmit and the backscatter signal will be recorded every 20 micrometers in the vertical direction. The blue line represents the first scan and is considered a reference scan. The green and the red lines are the second and the third scans. As time goes on, the backscatter signal increases at the bottom because the particles settle down, and more particles scatter at the bottom, while the number of the particles decreases at the top, resulting in a decrease in backscattering. After the measurement procedure, I will make an introduction to typical spectra of sedimentation, creaming, flocculation, and breaking. The first one is sedimentation. Sedimentation refers to the falling of suspended particles. Here I present delta transmission and a delta backscattering spectra of sedimentation. As time goes on, more and more particles scatter at the bottom, resulting in an increase in backscattering and a decrease in transmission at the bottom. At the top of the sample cell, the trends of backscatter and the transmit signals are totally different. The concentration at the top becomes lower and lower, so the intensity of the backscatter signal decreases, and the transmit signal increases. In this slide, I present both transmit and backscatter signals. However, when the stability analysis is performed, users only need to focus on one of them. In most cases, only one spectrum can be clearly displayed and completely characterize the unstable phenomena. Here is the measurement result of a titanium dioxide suspension. The concentration becomes lower and lower at the top because the particles settle down. the upper part of the suspension becomes transparent. Therefore, the transmit signal increases and the backscatter signal decreases. However, there is no variation of the transmit signal at the bottom, because the bottom part of the suspension is almost opaque even if the particles are evenly dispersed in the suspension in the beginning of the measurement. The backscatter signal shows two characteristic peaks for the bottom and the top of the suspension. So for analyzing the titanium dioxide suspension, the backscatter signal is preferred. Sedimentation is one of the most common unstable phenomena. It can be observed in slurry, pigment, vacuum, and suspension. The next one is the typical spectrum of creaming. The radiation trends of the signals are exactly the opposite of those of the sedimentation because particles or emulsion droplets scatter at the top instead the bottom. Therefore, the backscatter signal increases at the top and decreases at the bottom. Here I have an example of creaming in actual application. The sample is milk. As we can see, the transmit signal is not ideal because the sample is not transparent. In this situation, we need to focus on the backscatter signal, which increases at the top and decreases at the bottom, just as the same as that of the typical spectrum. Creaming often occurs in milk-based beverage, emulsion, oil, and pesticide. Flocculation, coalescence, and also ripening often lead to particle size variation. When we analyze the spectra, if there is an overall increase or decrease in the middle without characteristic peaks at the top and the bottom, it can be inferred that the particle size changes. As I mentioned before, the transmit and the backscatter signals change as the particle size changes. Whether the overall increase or decrease of the transmit and backscatter signals occurs depends on the size of the particle aggregates. For example, if the aggregates that form are at the micro level, the backscatter signal increases. If the aggregates are at the submilliliter level, the backscatter signal decreases. Here is an example of flocculation. The sample is wastewater with polyaluminum chloride, and the suspend particles started to coagulate and then flocculate. Flocculation leads to large aggregates, whose particle size can reach almost a milliliter. According to the change curve of transmit signal over particle size, the transmit signal will increase. Particle size variation often occurs in cowling, ink, wastewater treatment, and nanoparticle system. The last typical spectrum is breaking. Two or more phases will be formed when breaking happens. If only two phases are formed, the typical spectra are shown on the left. Suppose the density of the suspend phase is larger than that of the continuous phase, and the suspend phase tends to coalesce. They will settle down and form a new phase. The lower part becomes translucent, so the transmit signal decreases. As the concentration of the suspend phase becomes lower and lower in the upper part, the transmit signal increases. It is the same reason for the changes in the backscatter signal. Finally, the signals sharply change on the interface of these two phases. These spectra demonstrate how breaking occurs. The transmit signal is hardly detected, so we use the backscatter signal. Along the scanning direction, we can see a sharp drop in the backscatter signal, which is the interface of these two phases. Breaking often occurs in tofu, yogurt, beverage, and oil. In addition to stability analysis, mean particle size measurement can be carried out as well. The BeScan Lab offers particle size analysis based on three methods, namely gravitational liquid sedimentation, transmission, and backscattering. The gravitational liquid sedimentation is suitable for large particles that tend to settle down. The transmission method is suitable for clear, diluted dispersions with small particle size. The backscattering method is suitable for dispersions with small particle size, proper concentration, and a strong backscattering. If we want to finish the calculation, additional parameters related to the dispersions are also necessary. Users can check them online, or users can search them in the database of the software. The next part is the result analysis. When we analyze the sample, in most cases, it can be noticed that chaos signal often occur at both ends. This chaos signal is mainly caused by push-up, meniscus, and a sample cell. Therefore, it is quite important to remove these chaos signals to obtain the real signals that are related to the sample. Such signal processing can be carried out with the software. One of the most important functions of the software is quantification of destabilization using IUS, the instability index. The calculation formula is shown in this slide, which accumulates the variation between two consecutive scans, and we have experienced values that are related to stability. If the IUS is equal or smaller than 0.5, that means the dispersion is quite stable. An unstable phenomenon cannot be observed with our naked eyes. If the IUS is larger than 10, the dispersion can be considered unstable. Phenomena such as sedimentation or creaming occurs quickly and they can be easily observed with our eyes. The larger the IUS value, the more unstable the dispersion will be. We can compare the instability index curves of different samples. If the instability index changes slowly or the curve becomes smooth over time, that means the dispersion is stable over time. In this figure, the origin sample is the most stable, while the blue one is the most unstable. With the instability index curve, long-term stability can be predicted. The software provides a radar chart where the regional instability indices are illustrated, which facilitates the comparison of the instability indices of individual parts. The software identifies the sample to three parts: the bottom, the middle, and the top. On the left is an example. The sample shows a high instability index at the top. However, the general instability index is small, which demonstrates a stable dispersion with a relatively unstable top. Particle migration can be studied when sedimentation, creaming, or breaking occurs, because these phenomena are caused by particle moving. I will use sedimentation as an example to explain how to obtain the particle migration rate. These are two characteristic peaks in the spectrum, one for the opaque layer and one for the clear layer. First, we can use the software to limit the range of the characteristic peak. Here we use the characteristic peak that corresponds to the upper clear layer because this is the area where particles start to settle down. With the help of the software, we can get a change curve of peak thickness over time. By limiting the starting point and ending point of particle migration rate can be obtained. Besides, mean particle size calculation based on the gravitational liquid sedimentation method can be realized. Temperature is an important factor affects the stability, especially for the temperature-sensitive material. The BeScan Lab provides temperature trend testing, allowing the sample to be analyzed under different temperatures. In regular measurement, the color band on the right represents the scanning time. However, it represents the scanning temperature in temperature trend testing. Users are allowed to scan the sample under different temperatures to find out the change point. A sample can be analyzed in high or low temperatures. For example, agrochemicals are often analyzed over 15 degrees Celsius to observe their stability under high temperatures. The examples in this slide shows the change point of the sample is around 33 degrees Celsius because after the change point, the instability index increases rapidly. In the last part, I will show you some application cases related to stability analysis. The first case is beer. In this case, we investigate how the beer foam changes over time. Customers always associate the beer foam with the beer's quality and freshness. Besides, beer head provides an attractive look, so a stable beer foam is quite important to the manufacturers. In this case, 6 commercial beers with different alcohol contents and wort concentration were measured. This figure shows how the beer foam changes. As time goes on, the foam recedes and the water droplets between the bubbles drain to the interface between the liquid and the foam due to gravity to form a new drainage layer. The variations of transmit signal concerning to the phenomenon can be detected. As time goes on, the transmit signal shifts to the left on the far right side, allowing more light transmission because the foam breaks. The formation of the drainage layer allows more light transmission as well. We can also notice that the transmit signal of the liquid increases because the suspend substance, such as yeast cell and proteins, settle down. Here is the change curve of the peak thickness over time. The larger the drainage layer thickness in the same period becomes, the faster the foam will recede, and the more unstable the foam will be. High alcohol content is not beneficial to foam stability, as sample 4, which has the highest alcohol content, also has the thickest drainage layer. According to the analysis results of sample 4, high wort concentration can be regarded as an important factor that affects the stability as well. The reason for the unstable foam in sample 1 is low wort concentration. For sample 2 and sample 3, they have the same alcohol content and the wort concentration. However, their analysis results are not the same. The packaging foam is probably a factor. It seems that the beer foam of the bottled sample 3 is more stable. The next is the instability index curve over time. The greater the instability index is, the less stable the dispersion will be. Each scan provides an instability index, and after the measurement, the change curves of the instability index will be compared to each other. The instability index of sample 4 is the greatest throughout the measurement period, followed by sample 1, which means sample 4 and sample 1 are relatively unstable systems, corresponding to the measurement results using hydrodynamic analysis. Sample 5 and sample 6 with low alcohol content and proper wort concentration are relatively stable systems since their instability indices change slowly. The next application is ibuprofen. Ibuprofen is a suspension in which active pharmaceutical ingredients should be evenly dispersed. We have also measured their zeta potential and particle size distribution with the Binano and Bettersize 2600. The results are listed in the table. The sample 3 has the highest zeta potential and the sample 1 has the smallest mean particle size. Here are delta backscatter signals for the 3 samples. In general, sample 1 is relatively stable among the 3 samples because only creaming occurs. The backscatter signal decreases at the top of sample 2 because small particles settle down. The particles continue to settle down, resulting in an overall increase of backscatter signal in the middle. The backscatter signals decreases at the top and increases in the middle of sample 3, leading to breaking. According to the instability index curves, sample 3 is the most unstable even if it has the highest zeta potential. The sample 1 with the smallest particle size is the most stable. Stability is strongly associated with particle size, so the evaluation of stability can not only rely on zeta potential measurement. The instability index is a more comprehensive solution to stability analysis. At last, we measure 4 types of zirconia slurry. They are both used in 3D printing. As we know, uneven dispersion of oxidized zirconium particles may lead to structural defect in the finished part in 3D printing. Therefore, it is critical to analyze their stability. The zeta potential and the mean particle size of the 4 samples are measured with the binano. The zeta potential are quite high. They are all around 60 millivolt. Here are the delta backscatter signal for the 4 samples. Backscatter signals of the 4 samples indicate slight sedimentation, and the backscatter signals in the middle part slightly increase due to flocculation. Stability indices for 4 samples are all below 0.5, which indicate very good stability. In this case, result of stability evaluation using the BeScan is the same as that using the binano. At last, I will make a summary. We can use BeScan to analyze the stability of the dispersions. And we know that many factors can affect the stability, such as particle size, density, zeta potential, and the temperature. If the results of the stability analysis is not so good, we can use other Bettersize instruments to find out the problem. For example, we can use the Bettersize analyzer to find out the relationship between the particle size and the stability. And we can also use the binano and the Betterpick to investigate how the zeta potential and the density affect the stability. In general, Bettersize offers a comprehensive solution, not only the qualitative and the quantitative stability analysis, but also the ability to optimize your product. In the last slide, we present the specifications of BeScan. That's all for this video. I hope it is useful for you. Thank you for your watching.